nucleus, and surrounding them are cytoplasms. And again, only eukaryotes have a nucleus, as what distinguish these cells can classify in eukaryotes or prokaryotes, or you just pay attention about nucleus. If they have nucleus, they belong to eukaryotic cells, and if they don't, they belong to prokaryotes. Now, eu is mean two, and karys mean nucleus. Two nucleus, they have nucleus, so they belong to eukaryotes. And it means that having nucleus is defining features of eukaryotic cells. So in these lectures, I only mean nucleus. It means nucleus of eukaryotic cells. What you see here is nucleolus. I explain a little bit. Why is nucleolus? So in previous lectures, I teach you about Nucleolus, right? It's a singular form. We have Nucleolus, singular form. That's when is more than two Nucleolus, we have nucleoli. Okay, so it's the same, like we have a nucleus here, but in the plural form, it's become nuclei. Okay. In the single form, they are nucleolus. So ours become I, us here become I. One nucleus, two nuclei, one nucleolus, two nucleoli. Just singular and plural forms. Pay attention, because in the next slide, sometimes you see nuclei, sometimes you see nucleus, sometimes you see nucleolus, sometimes you see nucleoli. I don't want you to misunderstanding this term. So what could be the advantage of having a nucleus? So you can see here, in the prokaryotic cells, the transcription and the translation happen stimulously. It means they happen at the same time, and they happen into the cytoplasm because they don't have nucleus. In eukaryotes, you can see that transcription is inside the nucleus and translation in the cytoplasm. So they elaborate, the processing takes place to produce mRNAs from precursors RNAs, and the processing step complete before the mRNAs go out of the cytoplasms. Many macromolecules must be transported between the nucleus and the cytoplasm. We have mRNAs, exporters, we have our rRNA, tRNAs, ribosomes exporters from the nucleus also. So having nucleus allows cell to have much more complicated regulation of the gene expressions is possible in prokaryotic cells. And the second, eukaryotes normally they contains more DNA than prokaryotic cells, and in some cases it's I don't know, it's 10,000 times more. So the nucleus can protect the DNA of the cells or better. And can you guess the number of nucleus that a single cell have? So in these pictures, we have two nucleolus inside one nucleus. Can you guess the numbers of nucleus inside a cell? How many? Just one?
Depends.
It depends.
Depends on the types of cells.
Depend on the cell type. Depends on the functions, right? Remember in the first lesson when I talked about the fundamental units or of the living organism. When I show you the cells difference in the shape or the size or the structures is all because they have different functions. So, can you guess or can you give any examples of or the cell have multinuclei, the cell have no nucleus, and the cell have just one nucleus?
Red blood cells usually don't have nucleolus.
It's not nucleolus. Nucleus.
A nucleus.
And we cell have or many nuclei. So, the nuclei varies appearance according to the cell types and organisms. So they range in size from 1 micron to more than tens or micrometers in diameter. And the smallest or nuclei or the smallest nucleus they are approximately or 1 micrometer, and they found in in which living organisms they found in the yeast cells, Saccharomyces cerevisiae, the main. And here, most of cells have a single nucleus, but some cell contains multiple nuclei, and few cells lack of nuclei. So they can vary. Okay. In these pictures, we show in embryos of Drosophila at multinucleated stage. So a multinucleated cells can form when a cell undergoes nuclear divisions, because it carries or karyokinesis many times without undergoing cell divisions. And this can allow the RNAs and proteins produced in a subset of the nuclei to move through the common cytoplasm to set up the gradients. And in this case, they plays a central role to the development of the anterior and posterior axis of the flies. So, I will show you examples of a cell type which have numerous of the nucleus. They are major muscle cells. So in muscle cells, they are really big ones. And can you tell me why the muscle cells they need more nucleus than the other?
Is it because they use a lot of energy?
They use energies? Yes.
They use a lot more energy than other cells compared to.
Okay, I agree. They in charge or play important roles in our movements, right? So they need more energies. And when they need more energies, why do they need more chromosomes?
To produce more. I remember the name of the organism, but there's an organism that produces a lot of energy for the muscles, and I think the DNA, the more the DNA they have, the more they can produce of those organisms, or the small organisms. They can produce more energy for muscles, but I don't remember the name of it.
Mitochondria.
Yeah,
it is mitochondria.
Oh wait, no, is it mitochondria? Like, it's not produced by the DNA. It's more of symbiosis, no?
Yes.
So it's not like completely related to the nucleus normally.
But Nguyễn Đức Huỳnh,
the much bigger, so it need more cells, so the nucleus can divide to make the muscle bigger.
they divide, make the muscle bigger.
Yeah.
Actually, I mean that the muscle cells, they're really big. And they play important roles in our movements. And for your friend said that just because they need more energies, right? So they need more nucleus. And my question is why they, or when they need more energies, why they can or increase the numbers of the mitochondria. You know that the mitochondria is the energy factories for the cells. Why they just, or if you want more energy, you just increase the numbers of mitochondria. Why you need to increase the numbers of nucleus?
Because mitochondria have a
May I, Professor?
Yes.
So I think because to build new muscle cells, because we are in constant need of new muscle cells, we will have to keep up with the protein demand that the new cells division process require. Therefore, we need more nuclei to produce more mRNA.
More mRNA.
And basically to translate into protein.
So it's just about the proteins. What about the energies?
Well, you need protein for the production of mitochondria also. And because we need more energy, we need more mitochondria, which can be created by using even more protein.
Mitochondria produce more proteins?
No, but I meant that, no, I meant that
okay,
we need more protein to produce more mitochondria.
Do you know that mitochondria can multiply by themselves?
Yes.
It not depends by the nucleus. They can divide by themselves.
Can I answer your question?
Yes.
All
Lê
right.
Hoàng Minh Nhật, right?
Lê Hoàng Nhật Minh.
Lê Hoàng Nhật Minh, sorry.
So actually, I think the ideas of Winton is quite right, but the way he explained is not perfectly right. So the muscle cell needs more DNA, not because they need more to produce more protein, but because they need to produce like more cell instead of produce more mitochondria. Because if the muscle cell produce more mitochondria, they can cause overheating, and this can cause like the self death of themselves. So they choose increasing the number of muscle cells is a safe way to increasing the energy instead of increasing the number of mitochondria. I think so. Overheating is quite a dead cause, a dead ripple to the self.
It's not actually related to overeating. So we need to provide a lot of sugars for your muscle tissue for work. It's not about the overeating. And I say that the muscle cells is completely very big, and they're very long also. And they are skeletal muscle cells. They are formed by fusions of many cells. So many cells fuse together to make the muscle cell. Okay, so just why they become bigger and they become very long. And because of the syncytium conditions, tình trạng hợp bào syncytiums, Tức là rất là nhiều các tế bào nhỏ hợp lại, fuse together to make a very big master cell. Okay, so it's because they are very long. To control such a very long cell with many structures and function units, or just one nucleus might not be efficient. Cells, or you can imagine that your master cells, they are quite long and big like this. It's more like the same here. So they have numbers of nuclei inside. If just one in the center like this, imagine that it cannot work efficient. Cells, or the fuse numbers of the small cell to comes a big master cell here, and they still keep these numbers of nucleus inside to work more efficient, and they can control the long cell better. Okay, and the second things, and I agree with the first students, because they are master cells, they need enormous amounts of energies and also the proteins. So these energies is produced in many mitochondria inside them. So if I ask you a question, in which cell type we can find numbers of mitochondria, the answer must be the muscle cell. Okay, and some of the mitochondria, if they want to work, they need genes are in the nucleus. So they are the control centers. If they need the commands from the nucleus. So for example, here, if you have a small mitochondria here, it's very far from the nucleus in the center here. So they will receive the message is very slow. It takes a long time to send the message from the center to the nucleus here. So that's why to increase the speed of transfer the commands from the nucleus, we need more nucleus. One nucleus cannot fulfill the requirement of many mitochondria in very short time. Okay, so to make the energy production fast, the mitochondria needs more produce of those genes in nucleus at a very fast pace. Okay, so to make it fast and efficient, many nuclei are there. And the here requirement of proteins also cannot be met by one nucleus. Therefore, maybe nuclei in order to perform the transcription and translations for the synthesis of protein in very fast and smooth ways. That's the reason why we need more nucleus inside a master cell like this. Understand? It's okay for the answer. And in a contrast ways, we have fields or cells or one students have the examples of the RBC cells, red blood cells, and we have some other natural or blood cell type, and they are lack of nucleus. So in the case of the RBCs, because they in a biconcave shape, so it's critical for them to easy pass it through the tiny capillaries. Okay, so they don't need any nucleus. So they can move through our bloodstream and also the pressure on the blood vessel. They just go around your bodies and brings oxygen and oxygen. Okay. For the next, I will introduce about the major compartment of the nucleus. Is maybe or in the previous lesson you have the imagination about the nucleus. They have first the nuclear envelope or the nuclear membranes. They are double membranes. And on the surface of the nuclear envelope, we can find numbers of the nuclear pore. We call them NPC, nuclear pore complexes, numbers of nuclear pore here. The second part, they are nucleolus. in the plural form, nucleoli. And their way the ribosomes will form. Okay, so they have the RNAs or rRNA synthesized, and they assemble to make a mature ribosome. And the other part is nucleoplasms. They are non-nucleolar regions of the nucleus. So we have nucleoplasms inside nucleus, and we have a cytoplasms outside of the nucleus. And in the previous lessons, I told you that if nucleoplasm plus the cytoplasm, we have protoplasm. And the most important part inside a nucleus, the thing that controls all the activities inside a cell, we have a chromatins. So chromatins, they consist of a double-strand DNA complexes with histone proteins. And they have two types of the chromatins. Heterochromatin, they highly composite. And you can see the dark part here, the dark part. Oh, sorry. I'm sorry, I have a phone call, so I just out of the meetings. So you can see here in, can you hear my voice?
Yes, but we can't see the
Okay.
screen.
Yeah,
the screen.
You haven't saw your screen.
All right, I haven't set my screens. Hello, it is work now?
Yes, it works now.
Yeah,
We can
yes.
see the screen and hear you.
Okay, sorry, we keep continuing. Really sorry for the this stuff. And here you will have the heterochromatins. You can see the black here. They are highly compacted and they have a ear chromatins in a more slight here. They are less dense compacted. And for the heterochromatins, they contain genetically inactive satellite sequences. So for the nucleus envelope, they have a double membrane. The outer membrane is continuous with the membrane of the ERs, and the lumens of the nucleopores is continuous with the lumen of the ER. For this is not easy to see, so I have to write it down. We have a membrane here. For example, we have a membrane of the nucleus. This is the inner membrane. And they continuous with the outer membranes like this. And also, you know what it is? The purple ones.
The Endoplasmic Reticulum.
Yes, they are ER. So we have inner membrane. We have outer membrane. And we have rod ER. Okay, so that's why the outer nuclear membrane is continuous with the membrane of the ERs and the lumens. Here's the lumens between the outer and inner membranes. The lumens is continuously with the lumens of the ER. And they also contain numbers of the NPC. So we will have the NPC here, nucleopore complexes. It's like a door, and it's allowed or the compounds go in or out of the cells. They are door of the membrane here. They have numbers. Okay. So the NPCs, they are large molecule. They actively transport between the nucleus and the cytoplasm. With the uncharged molecules smaller than 100 dantons, they can pass through the nuclear envelope. And with the bigger ones or the charged molecules, this can go across the nuclear envelope. You know that's this is high school knowledge. Can you tell me the average molecular weight of an amino acid? What is the average molecular weight of amino acids? Các bạn khối B đâu rồi ạ? Khối lượng phân tử trung bình của amino acid là bao nhiêu?
110 Dalton.
Yes, 110. And in high schools is?
Sorry, can you repeat?
DVC, right? Nào, ở trong trường trung học thì mình học khối lượng phân tử trung bình của một axit amin là bao nhiêu nhỉ? 110 đơn vị carbon đúng không?
110.
Đơn vị carbon đúng không? DVC. Thì đơn vị carbon thì nó equal to dantons. Okay. So it's all dantons or đơn vị carbon như ở cấp 3 các bạn học ấy, they are the same. They are under alternate names for the atomic mass units. So one danton is equal to the mass of the single hydrogen atoms. Okay. And here's so it means that with the molecules is more than 100 dantons, they need to move through the nucleopore complexes. Because the average molecular weight of an amino acid is 110. So what about the average or molecular weight of the nucleotides? 300? Okay, so let's skip this. Here, these are the pictures under the light microscope about the membranes of the nucleus. So you can see that we have a numbers of NPC here. Just one dot is one NPCs, and they made of about 30 to 100 nucleoporins proteins. Many different class of molecules and macromolecules are transported through the NPCs. So the ions, because they are charged molecules, we have numbers of proteins or sufferings, and this is transported proteins. We have the RNAs, mRNA, tRNAs, and rRNAs is need to transport through the NPCs, and the virus need to transport through the NPCs, and also the ATPs, adenosine triphosphate, the energy money of the cells need to transport through the NPCs, and the big molecules like glucose needs to transport through the NPCs. Number of things need to transport through the NPCs. They are the doors of the nucleus. They control everything in and out. And the next, proteins are selectively transported into the nucleus through the nucleopores. We have here the major nuclear proteins contain the sequence information that requires for those nuclear localizations. And the signal within the proteins to get into the nucleus chest or of amino acids termed the NLS, nuclear localization sequence. So here we have examples. When they want to inject the nucleoplasm or into a cellulose oxide, the nucleus of cellulose oxide, and they have a surface or a case here. One nucleoplasm completed one with a tails and a core. Pentamerase mean we have a 5 units to make that ones. Or they have a core with only one tails, or they have it only tails, or they have it only core to index inside the nucleus of the cellulose oxide. Whenever these nucleoplasmids contain the tails, the sequence of the tails, they can go through the nucleus of the cellulose oxide. So it is prove the importance of the this kind of sequence, nuclear translocalization sequence. Whenever you have these kinds of sequence, these kinds of compounds can go into the nucleus. Okay. What about the export? If you want to export something through the nuclear envelope, you need another sequence we call the nuclear export sequence. And they can bind to the protein that's contain the NES, and they can transport them into the cytoplasm. And the NES and also NLS, they were discovered or in the course of studies of the growth of the human HIV infected virus. So in virus, they have a small proteins HIV, they call the rats. This is the keys for the HIV virus. They have rat proteins, and these kinds of rat protein, they contains NLS to import things inside the nucleus, and they contain also NLS to export something out of the nucleus. So the rat proteins, they can move out of the nucleus to the cytoplasm, where the RNA relieve and re-enter the nucleus for another route. Okay, here they contain NRF. Because they have NRF, they can go into the nucleus. And when they get into the nucleus, they can bring their RNA. So HIV virus are RNA virus. So they bring their RNAs by these kinds of proteins, and just because they have NES, they can go out to the cytoplasm. So they bring their RNAs successfully to the cytoplasm. That's the way they work. Okay. Các bạn hiểu không ạ? Khi mà cái màng nhân này nó rất là selective nhá, transport nhé. Nếu như các bạn có chỉ khi các bạn có một cái cấu trúc là NLS, một cái chuỗi protein như thế này, thì các bạn mới có thể, thì cái hợp chất này mới có thể đi vào trong nhân tế bào. Và khi mà các bạn có một cái cấu trúc là NES này, thì các bạn mới có thể đi ra, thì sorry, không phải các bạn, thì cái hợp chất này mới có thể đi ra ngoài màng nhân. Thì cái việc mà phát hiện ra cái cấu trúc NES và NLS này nó được phát hiện khi mà người ta nghiên cứu về virus HIV, virus HIV. Và ở virus HIV nó có một cái loại protein là cái rat protein này, nó mang cả NLS và NES. Và virus HIV là cái loại virus RNA, nó đã xâm nhập vào trong, nó đã sử dụng cái bộ máy DNA của trong nhân của chúng ta, nó sẽ nhân lên rất là nhiều các RNA của virus như thế này. Và nhiệm vụ của nó là bây giờ nó phải dùng chính cái protein của nó này, nó làm rất là nhiều vòng, nó chui vào đây, nó gắn với cái RNA của nó, và nhờ có cái cấu trúc NLS mà nó có thể chui vào, và nó có cấu trúc NES để nó có thể export ra, và như vậy là nó mang thành công cái RNA của nó ra ngoài cytoplasm. Và khi mà ra ngoài cytoplasm thì nó sẽ kết hợp với protein của nó và nó sẽ lắp ráp và tạo thành một cái virus hoàn chỉnh, đúng không? Đây là cái cách mà virus HIV có thể vận chuyển cái RNA của nó ra ngoài nhân. Và nhờ cái nghiên cứu này thì người ta phát hiện ra là muốn mang cái gì vào nhân, mình phải có NLS, và muốn mang cái gì ra khỏi màng nhân, ra khỏi nhân thì mình phải có cấu trúc NES. Okay. So, multi-classes of RNAs are exported from the nucleus. We have the mRNAs, tRNAs, and ribosome RNAs are produced in the nucleus, and they exported through the NPCs. And the same NPC used for protein transport are also used for RNA export. Export of RNAs is receptor. They require the receptor, and they require energy also. Right. This is one example of a theories we have. Transport of most RNAs in unidirectional from the nucleus to the cytoplasm. So when you inject the RNAs into the cytoplasm, even we have mRNAs, RNAs, tRNA, or USR or RNA, they are small RNA, they control the transcription. So you have one series, you have no more types of RNA, you have a small RNA here. And they're in charge of slicing the mRNA to make them to become the mature messenger RNAs. So when you inject numbers of RNA into the cytoplasm, they still into the cytoplasm. But when you inject the RNA in the nucleus, you can find numbers of them in the cytoplasm. So the transport of the most RNA are unidirectional. Okay, but until now we know that the tRNA can be imported back into the nucleus and subsequently re-export. So that's why I use here most of RNAs unidirectional from the nucleus to the cytoplasm. Just one way, unidirectional. We have another structures inside the nucleus. Actually, they support the nucleus membranes. We have a nucleolaminars. So they're located beneath the inner nuclear membrane, and they physically connected to it by laminar associated integral membrane proteins. They play roles in nuclear envelope assemblies and may provide physical support. And the protein complexes that interact with the nuclear laminar cross the nuclear envelope and link to the cytoplasm of the interior. So these two structures is the cytoskeletons into the cytoplasm. We have a structures beneath the inner nuclear membrane here. They are nuclear laminars, and they have also structure here to connect the nuclear laminars with the cytoplasms, and they will support the nuclear envelope. That's all. And the yeast and some other unicellular eukaryotes, they don't have nuclear laminars. It's just maybe they just because the yeast cell is very small, and because they are very small, so they don't really need the nuclear laminars to support the physical functions. And also in in their cell divisions, the cell or the nuclear membrane do not disappear, so they do not need any support from the nuclear laminars. And another, we have a nucleoskeletons. They have organized the nuclear functions. So we have a cytoskeletons, and inside the nucleus, we have also nuclear skeletons. Are these a little bit complicated? Or just imagine that we have a network inside the nucleus to make all the things or work smoothly. Okay, inside the nucleus, I don't require you to study this slide. Okay, so about the nuclear membranes also, we have the nucleus have double membranes. It means that we have another theories, endosymbiosis theories, the hypothesis, endosymbiosis hypothesis. They propose that these kinds of organelles arise during the evolutions when one cell endocytoses another cell and gets another cells. And the ingested cell would be then surrounded by two membranes, one of its own, here injected, one of its own, and another by the membranes of the engulfing cells. Is quite logical with the mitochondria and also protoplast, but for nucleus is not really, we don't have silence explained for them. Another structures inside the nucleus, we have nucleolus. They are the size of the RNA synthesis, and also they assemble the ribosomes. Okay. And another components, we have a chromosomes. Chromosomes, They are occupies distinct territories inside the nucleus. X chromosome occupies in a distinct regions, and they can prevent the chromosome from becoming entangled with another one. And the nucleus contains both chromosome domains and interchromosome region. So we do not know how chromosomes territories maintains, but many types of cell and the chromosome they call the telomere, or they can anchor to the nucleus envelope. So they're in the separated region like this. Or what is the telomeres? I will explain in the next slide. And now we have a videos or to summarize what we learned about the nucleus.
Here's a diagram of a eukaryotic cell. Let's look a little bit closer at the structure of one of the most important organelles in the eukaryotic cell, the nucleus. So here we have a depiction of the nucleus. And the most important function of the nucleus is to contain the genetic material of the cell. But what is the structure? Let's look at the outside of the nucleus first. The nucleus is surrounded by two separate membranes, an outer membrane and an inner membrane. Let's look at this structure up close. So we have an outer membrane and we have an inner membrane. These two membranes separate the nucleus from the cytoplasm, which is the liquid-filled space that makes up the majority of the cell and all of the non-compartmentalized parts of the cell. Essentially the parts of the cell that aren't enclosed within an organelle. On the inside of the nucleus is what's called the nucleoplasm. And the nucleoplasm is the fluid inside the nucleus. And so the cytoplasm and nucleoplasm aren't continuous. However, a lot of the times stuff needs to be transported between the nucleoplasm and the cytoplasm. So think the interior of the nucleus is where mRNA is produced, but mRNA needs to get into the cytoplasm where it can be translated into protein. And also, proteins in the cytoplasm need to be able to get into the nucleus, such as polymerases, which convert DNA into messenger RNA. So this transport of things like mRNA out of the nucleus and protein into the nucleus happens at a special complex called a nuclear pore. And so there's a nuclear pore over here in my blown up diagram of the outer and inner membranes. What's interesting about the nuclear pore is that it spans both membranes, the outer membrane and the inner membrane, so that compounds in the cytoplasm can be transported into the nucleus and compounds in the nucleus can be transported out. But now, the cell has gone to a lot of trouble to create these two lipid membranes to isolate the nucleus from the rest of the cell. So the nuclear pore is actually very selective in what can be transported inside and out. And so the nuclear pore actually recognizes special signals on different proteins, and only with the presence of these signals can proteins be transported into and out of the nucleus through the nuclear pore. So that the nucleus's primary function is to contain the cell's DNA or chromosomes. But when you look at the nucleus up close, you see there is a very densely compacted area. And this area is called the nucleolus. Just as the nucleus is kind of the center of the cell, the nucleolus is the center of the nucleus. But why is the nucleolus so much more compact and dense than the rest of the nucleus? Well, That's because the nucleolus has a very important function, and that is the site of ribosome assembly. So the nucleolus is densely packed with regions of DNA that produce not messenger RNA, but ribosomal RNA, which is the RNA that makes up the majority of the ribosome. And it is at the site of the nucleolus where this ribosomal RNA is assembled with the proteins that also make up the ribosome into a fully formed ribosome. And the ribosomes then can be trafficked through the nuclear pore out into the cytoplasm. The ribosomes also have proteins in them, and these proteins are produced in the cytoplasm and are trafficked back into the nucleus where they can be assembled and then make the return trip back into the cytoplasm. So there's lots of stuff moving back and forth in the nuclear pores, but it's very important for the compartmentalization of the nucleus from the rest of the cell. To move into our next discussion, the nucleus is intimately associated with another organelle, the endoplasmic reticulum, which you can see in the faded green outline. An interesting topological feature of the nucleus is that the outer membrane, remember which we've drawn up in the top left, is actually continuous with the membranes that make up the endoplasmic reticulum. So I'll just kind of draw that. So this outer membrane just turns around and actually becomes the membranes of the endoplasmic reticulum. I won't draw it all in. So the interior space of the endoplasmic reticulum is continuous with the interior space between the outer and inner nuclear membranes. And one final term used to describe the nucleus is the nuclear envelope. And the nuclear envelope just refers to the combination of the inner and outer membranes along with the nuclear pores. So these three structures together comprise the nuclear envelope, which encloses the nucleus.
So, or at the end of the first part of these lessons, or maybe we have 20 minutes break times, and we will back at or 11:30. Okay. The second part of the lecture today is about the chromatins and chromosomes. So inside the nucleus, we have genetic materials, so which will control all the activity of the cell. They are chromatins and chromosomes. So reminding you a little bit about the prokaryotic cells, the genetic materials in the form of our nucleolus. And in eukaryotic cells, genetic materials is the mass of chromatins within the nucleus. And here's a pictures of chromosomes. They are a pair of chromatids. And they stick together in the regions we call the centromere. So that's I use a lot of work about chromatins and chromosomes, and also the chromatids are. Who can tell me what different between them? What different between the chromatins, chromosomes, and the chromatids? I think you all learned in your high school.
Yes, well, chromosomes are composed of chromatin,
Yes.
which are then united with by the centromere.
Yes, they are dense. Okay. You think that they have a centromere?
No, it's just well, the place that they're connected is the centromere, like the chromosomes. No, wait,
so,
no.
What is chromatins?
The chromatids are the two things that compose a chromosome, I think. are the two parts of the chromosome. And normally they're condensed to make the image that you were showing to make a chromosome. And those two chromatids are connected through the centromere.
When we have the chromatins.
Well, the chromatin is normally, well, the chromosome, it's decondenses itself into chromatin when we have, I think, the cellular division, or when there's a reduction of the DNA. And then it is recondensed into the chromosome after the division is finished.
Is our almost true? Okay, so we move to the just for the different levels of DNA packaging. You know, that's chromatins is made of DNAs and also the proteins. So we have a different level of DNA packaging here. The diameters of a DNA double helix, we have a 2 nanometer. And when they turn around with a okay, Tâm Dan, okay, you can leave. Just send me an email, okay? So the second level is when the DNAs turns around the proteins in yellow here, they make a form of chromatins with 11 nanometers. And these kinds of chromatins pack together and they make a structure we call the nucleosomes with 30 nanometer in diameter. And for the next, when the section of chromosomes extended form, they fold so many times here, they make structures with 300 nanometer. And when they condense, we only observe the chromosomes in the cell division stage. And when they condense, they form the chromosomes and the diameter of 100 nanometers. So the DNA molecules has been packaged into a meiotic chromosome that is 10,000 fold shorter than its extended type length. So this is the pictures of the 30 nanometers selenoid structure fiber forms in a light microscope. So this is the in condensed form. So, and this is the decondensation form. So in the decondensation forms, they will allow the transcription process occur. And here's for the nucleosomes here, 30 nanometers, we call them the subunit of the chromatins. These contains 200 base pairs of the DNAs, 200 lengths, and 2 copies of the core histone. We have H2As, 2H2As, 2H2B, 2H3, and 2H4. They make the optomic cores. So 8 particles to make one thing. We have optomic core. And here's 2H2A, 2H2B, 2H3, 2H core is make the protein histones, and all the DNA's wrap around them is make the nucleosome here. And between these kinds of nucleosomes, they have one's linker histones. And the linker histone, they bound to the wrapped DNAs around the histone optomic cores. And here's the DNA's 200 base form of the DNA's graph 1.65 times for this part, just remember for me, the name of the histone optomic core. Sorry. So just remember, we have 4 ties of the histone to make the optomic core. And without the S1, the linker histone only. And for the red, H2A, H2B, H3, and A4, they make the histone optomic core. May I ask questions? Do you know why the DNAs can graph surrounding the histone optomic core? Do they attract each other, or the histone optomic core have any charge, and the DNA have or what kind of charge? Remember, in high school, I think you have learned this. Why they can attract each other?
Is it something related to the the electronic charge?
Yes.
Or something like that.
So they have different charges,
yes, and then so they attract each other, and with the structure of the histones, well, it produces this thingy.
Can you guess the charge of the DNAs?
I think it's positive. No.
Positive.
And the histones are negative.
So why DNA could be the positive? So the structures of a nucleotide, we have sugar, we have base, and also we have the phosphate group. What could be the charge of the DNAs? Okay, or discover as homes to see, to define what the charge of their histones and also the charge of DNA. So why they could attract each other. Okay. So one is positive and one negative. Okay.
I think the DNA is negative due to the phosphate group.
Yes, due to the phosphate group, they have negative.
Yes, and the histone may contain some positive charge amino acids like leucine. No, I don't remember the exact name.
Glycines?
No, not leucine. Leucine is natural one.
Biscay? Sorry, okay, it's too far from this.
Lysine.
Lysines. And what else?
Asparagine or I cannot remember. Sorry.
You can remember it in the biochemistry in your second years. No, in the second semester. Okay. We don't have a lot of time, so we will move. Here is, or just pay attention here, the structures of the nucleosomes by a different side. So from this side, you have 4 ties of the histone here. And this side, you can see the DNAs graph around the histones. Okay, so we have the H1, the histone H1 is linked here, the linker histone. And for the next, the chromatins remodeling complexes, decondense and condensation they need energies. They have a remodeling complexity, and they require the energies to make the condensed chromatins into the decondensed chromatins. So it is in our active state, right? So we have a transcription in this state. If they are condensed, nothing happens. And also, chromosomes have bending patterns. So the certain staining techniques cause the chromosome to have the appearance of the series or striations we call the G bands. The G band is the area that contains high level of G of guanine and cytosines. And for the next structures of the chromosomes, we have a telomere. Are in the centers when if you have another chromatins here, they will stick together. Here we have a centromere, right? But the end of the chromosomes, we have a telomere. The telos is mean and the is near here, but is mirrored. It's called the part. So together we have telomere is the end part. The end part of the chromosomes, we have a telomere. How is important? The parents who answered my previous questions, can you tell me what is a telomere and the function of a telomere? I want to give you some extra points. Do you know what is telomeres?
I think telomere is a repeated section of DNA that protect the chromosome from attack each others, and it will separate them.
Separate them.
If they don't have telomeres, they can attack others.
It's not really. But in the previous slide, I showed you that by the telomere, they can keep these kinds of chromosome in a separate area, right? We have the separate area for the chromosomes inside the nucleus. In the previous slide, do you remember? So, for the next region of the telomeres, for the telomeres, it's actually the repeated sequence of the nucleotide here. X and the chromosome, we have a telomeres. So, in each time the cell divisions, the telomeres will be shortened. And when the telomeres has been short enough to delete, the chromosome will be damaged. It will be break down or no more or exist in your cells. And who can tell me why each time of divisions, the telomere will be shortened? Shortened after each time of the cell divisions. Why this could be shortened?
Because the DNA polymerase cannot replace the nucleotide at the 5' and
right?
yes, it cannot replace it with nucleotides. So each time the cell divides, the DNA gets shorter because that part doesn't get replaced with DNA nucleotides.
Why? Can you explain more about the when?
Okay, so the DNA polymerase, the enzyme that synthesizes DNA during cell division, it can only add nucleotides to a existing 3 prime. Yes. So so the synthesizing DNA is direct. It's only one directional. So the DNA polymerase cannot replace the starting point of that new strand because it doesn't have a RNA strand before it, so it will get replaced.
It's replaced.
No, it cannot.
Is almost correct, but I expect to hear from you the primaries, the Okazakis, right? Do you remember when we talk about the DNA replicates? Remember that us? So okay, you can answer it as home. So it's correct, but it's not enough for me to give the extra points. Okay, so we have a telomerase, another kind of enzymes. So each time the cell divisions, the chromosome will be shortened a little bit. So it requires the telomerase. Here is one kind of enzymes. They have a T parts from proteins, and they have a third part containing the template region here is made of RNAs, and they are reverse transcript RNAs or enzymes, and they can allow the telomerase prolongs after the cell divisions. When we have telomerase, the telomeres will be maintains the length after the cell divisions. Sorry. And by the telomerase, so the chromosome will maintain the length. And is the discovery of the telomeres, and the telomerase will give the 3 scientists here the Nobel Prize in 2009. So in the first person we have Elizabeth Blackburn, and she found a N sequence in the chromosome of the tetrahymenars. I don't remember exactly the names of these organisms. Tetrahymenar. They are the single cell. They are single cell organisms that live in waters. So she found that the chromosomes of the tetra of this of these organisms, they have small calf at the ends here, end parts. They found the end parts of these single cell organisms live in water. They are the sequence that repeat the DNA rich in guanine. So numbers of guanines here, GG, we have GG or AT and GGGG, like this, repeated DNA rich in guanines. And in the same years, we have the second scientist, Shostak. He created a mini chromosomes, artificial mini chromosomes. So synthesis by the human. Okay, mini chromosome nhân tạo, artificial mini chromosomes. He creates one. In the same years, Elizabeth Blackburn found the end parts of the chromosome of another organism. And when the Jax inserts these or his mini chromosomes inside the yeast cells, when he inserts these artificial mini chromosome in the yeast cells, it has been degraded. It's no more, or you cannot find these kinds of the mini chromosome here. But when they combine together, these two kinds of scientists found others, and they decided to take the cap region of the Elizabeth Blackburn file here, Cooper, with the artificial mini chromosomes of the Jack's discoveries, and then they transfer the gains into the yeast cells so they can stay here. They are stable inside the yeast cell, and they have conclude that these kinds of telomeres, these kinds of end parts, they have played in very important roles in the protect the chromosomes inside cell. And there are current writers, sees the students of the Elizabeth Blackburns, sees the ones who found the telomerase. Any times when the cell divides, it will be shortened a little bit. And when the cell have the telomerase enzymes, it will prolong these kinds of end parts. So can you guess or the roles of the telomerase and also the telomeres are inside cells? What happens if the cell have to maintain these kinds of telomeres? When they have the telomeres, do we exist forever? We are immortal ones, right? X cells, when the cell divides, it will be shortened a little bit here. It will be shortened. So, or in normally after 30 to 50 times the cell divisions, the cell will be damaged. And they will be days when the telomeres end. They suicided, they kill themselves because they have no more telomeres. They divide enough time. So what happens if one cell have these kinds of telomeres after the cell divisions? They can divide so many dividings, so it can causing the cancer cell, right? The cancer cells. So, and they discovered in cancer cells from 95 to 85 to 90%, the telomerase is active in the cancer cell. So that's why the cancer cells can keep dividing forever and they make tumors, right? Very big one. They never kill themselves because they have a telomerase, that they have a mutation in the telomerase.
Recording in progress.
Treatments to cure the cancer patients, right? So they received the Nobel Prize for their discovery. Oh, here's why telomerase matters. So in cancer, they can multiply so many times and they make a big tumors here. Right, the tissue renewers, or more or less the same. Okay, for the next, in the humans, we have a 23 chromosome pairs to make our human genomes, but some maybe have the abnormals of the human genome, and they resulting in some kinds of or in genetics or disease, like we have a Down syndromes. Remember? So this is the pictures when we are in the fluorescent in situ hybridizations, we staining the chromosome with different colors, and we can make them in orders. We have 20 pairs of them. And the pictures of the human chromosome here, we have the karyotype. Karyotype is the pictures of the human chromosome. So, and two or closely related species can have similar genome in size, but they vary different in the chromosome number. Here's two kinds of the Manjaks from Chinese and from the Indians. The evolution from the Indian Manjaks, deer, this is one kind of deer. The deer from the Indians thus varies from the deer from the Chinese. By the chromosomes were initially separated and remain separate in the Chinese, but during the evolutions, these kinds of chromosomes they stick together and they fuse together to reduce the number of chromosome pairs, but without having any major effects on the number of the genes. So they just fuse, but the number of the genes remain the same. So we have two species, one deer from Chinese deer, and also Indian deers, with difference in the number of the chromosome. And for the species with a chromosome sex determination, the sex of the individual parents, and interesting problem for gene regulations because the variations in the number of the X chromosome. Now we have a different here, the doses comprehensions. The equal levels of expression of the X-linked genes into the two or sexes. So if the X-linked genes were expressed equal in X sex, the female would have twice as much X products as the males. I let just some minutes for you to discuss for the midterms. We will have the attending checks now, and also I left you some more minutes and I'm waiting for the question from the previous lectures. If you don't understand in any points or any points of the midterm, you can ask me, okay?